Next up, we have Oisín Biotechnologies, and apologies for the late start, but we are very excited to see what you're working on.
So yeah, my name is Matthew Scholz. I'm the CEO of Oisín Biotechnologies, and we're a genetic medicines company focused on longevity. And so I'm going to tell you a little bit about kind of what we've built and what we're doing, and share a little bit of data that I hope is relevant to this audience, and maybe ideally even can inspire some ideas for collaboration with the people who are here in the room. And so I guess to frame things, we started going after effectively aging itself, or trying to address it more mechanistically. And the reason we've done this, and I'm sure a lot of you have seen graphs like this in the past, but aging is pretty much the master corollary for any malady you could dream up. You have worse odds of having cancer as a clean living 70-year-old than you do as a chain smoking 20-year-old. And this isn't to say that your ill-spent youth and bad choices won't haunt you later. It's just that aging itself is such a dominant risk factor that it dwarfs out the current activity. And so we wanted to attempt to address this more mechanistically instead of going after the symptoms of it. And I'll start with something that's probably a little controversial. I don't think that we're ever going to get there with pills. And it's not to say that we haven't gotten mileage out of small molecule medicinal chemistry, or even that there aren't people in this room building new molecules that'll be useful. But messing with the chemical substrate of life, I think, is kind of fundamentally flawed when you look at it in the context of aging. That life is based on information. The essence of life is your genetic code, and in monkeying around with the chemistry is a little bit like, say, trying to debug Microsoft Word by changing the microchips in your computer. It's not that you couldn't do it, but it's going to be fraught with unintended consequences. And I mean, we see this every day, and like any drug you buy has a list of side effects longer than the intended effects. And so we wanted to attempt to address aging specifically at more of a genetic level. And this is I suppose, not entirely controversial on its own. And in fact, a lot of new drugs are even being discovered based on their genetic underpinnings. But it's been a challenge that, I mean, your body has a vested interest in not letting you monkey with its DNA. And we have these tools that have been the workhorses of the field for the last couple of decades, kind of fall into these two different camps of like viral-based vectors and nanoparticle lipid-based vectors. And they each have kind of strengths and weaknesses. Like a virus is a purpose-built machine designed to deliver code to life. I mean, that's what they do. There's been viruses, as far as we know, for as long as there's been life. But they also are kind of known bad actors. Your body doesn't like them. They're expensive to make, of course, also, and you can't usually redose them because you effectively vaccinate yourself against the vector. And whereas the lipid nanoparticle type technologies, I think everyone is probably quite familiar with now from their kind of use in the COVID vaccines, Pfizer and Moderna's in particular. And these have some advantages in that they're relatively easy to make at scale, relatively inexpensive, and they're not viral, so they're not really immunogenic on their own. But they have significant biodistribution issues, and I'll show you some kind of gruesome data for this a little bit later. They like to accumulate in the liver where they can cause lots of damage. And so what we set out to do is, like, from a tools perspective, was build a new kind of platform that combined some of the best attributes of both of these. And for those who aren't familiar with it, it's called a Proteolipid Vehicle, or a PLV for short. And I'll spend a couple minutes introducing it, and then I'll kind of shift gears into kind of what we're up to with it. But it's already been used in humans. It's actually just, I have interim data for a phase 2 clinical trial and COVID vaccine for it. And it was the subject of a very large licensing deal with Eli Lilly and Entos, our sister company, at the end of last year. Is, I believe, one of the largest delivery deals ever done. And so this talk and our applications are a little more, I guess, translationally focused than academic. But the whole issue with those lipid nanoparticles is that they had to make a Faustian bargain of sorts with charge chemistry. So in order for these things to work, you inject them. They need to be endocytosed, swallowed up by cells, and then they have to escape that pathway and deposit their cargo into the cell. And in order to do this, they need to have these charged lipids, cationic or inducibly ionic lipids. And the problem is that when those reach high concentrations, they become very toxic. And if you think about it, like even in the case of the COVID vaccines, the maximum dose they could give to a full-grown man was 250 millionths of a gram. And I mean, that's a really small amount. And putting in more would likely have had benefit for efficacy, but it was toxic enough that it toxed out the sentinel patients in the trial. And so the real challenge with this modality has been much more on its tolerability than its efficacy. And so we want to remove those, but if you remove those toxic lipids, then you don't have a way into the cells. And so we make use of this fast protein to get in. And it's kind of an interesting history. It came from something called a reptilian Ortho Rayovirus. It's a virus that normally infects the stomachs of alligators. And it's unique in that it's the only non-enveloped virus known to science that has a fusion protein like this. And it's also unique in that it's super, super tiny. So you can see it here next to the spike from influenza. And the virus actually doesn't use it to get into cells. It uses it to fuse adjacent cells together into these syncytia. And but because it's so small and it kind of hides out just in the lipids of the particle, it's effectively invisible to the immune system. And so what happens when you have a particle with this on the surface, like the one we've built, when it gets close to the cell, it forms kind of a little complex and it mixes the lipid of the cell with the lipids of the particle. And so it never needs to be endocytosed. You can block endocytosis, it works fine. You can chill it down to 4 degrees, it works fine. And it's effectively indiscriminate delivery to any cell you want. And so this became the workhorse for all this technology I'm going to show you. And this, I guess, is a little bit more on the toxicity aspect. These are livers from mice, rather unfortunate mice, as you might gather looking at them. And we were basically comparing the PLV, our delivery technology, against a lipid nanoparticle, kind of like MC3 formulation, like you'd see in Moderna's. And what you can see is, it looks like someone took a blowtorch to the liver. And this, incidentally, was lethal within 24 hours to the mice. And I don't think actually the liver damage is what did it. I think what actually got them was this crazy cytokine spike, something called interleukin 6. So it's effectively a severe reaction to them when they're administered IV. And for context, this is ours at 20 mg per kg versus 5 with the LNP, and you can see there's effectively no damage on it. And in this case, you're getting this massive spike at only half a mg per kg. So it's way too toxic to use. And this has been demonstrated in clinic as well. There's a reason they don't do high doses of these things. But if you get rid of that toxicity, you can push the dose up much higher, and it allows you to deliver to all these tissues in the body that you couldn't reach before. So now you have a tool that can effectively deliver genetic information to anywhere in the body. And we've used this with a broad mandate to go after all sorts of aging-related diseases, from functioning initially as a senolytic, to getting rid of damaged cells, even in cancer, building collagens, like remodeling tissues. And so I'm going to highlight some that I think are more interesting for metabolic applications. But we gotten started using this tool to kill senescent cells. And I'll show you a little bit about how it works. It's cool, and it illustrates what the technology is actually doing. But the idea here is we wanted to get rid of a damaged cell based kind of on what it was thinking, not what it looked like. So based on its transcriptional attributes, not its surface markers. And so the idea is you could administer this IV. It could go throughout all the cells in your body, but it would only activate in a senescent cell in this case, or a cancerous cell. And we put a suicide gene under it that allowed us to make those cells just self-destruct. So you can actually watch them go into apoptosis with the time-lapse microscopy here. It's kind of cool. But what is more useful or interesting is that it can selectively get just those cells in a mixed population. And I think we have a lot of experience killing cells in the context of cancer, but the standard of care for cancer is effectively eating poison. This is not something most people associate with longevity, or it's definitely going the opposite direction as the last talk. And so the ability to pick off just one damaged cell in an ocean of healthy cells without collateral damage was a tall order. And I think really the only way you could solve it was to not use the poison at all. And so in this case, it's It's a little computer program written in DNA. And this is kind of just another illustration of this idea at work. We're making Firefly protein in this mouse, and you can see, like, if you administer it IV, the whole mouse lights up, all of its organs light up. But here on the right, this mouse got the exact same dose, administered the exact same way, but now we've got that Firefly protein under a genetic control that only turns on in tumors. There's a little tumor implanted in its flank. So you can see that even though all these tissues took the same dose, it didn't activate anywhere else. And this allows us to do some pretty exciting things therapeutically. And so the first one, so that's, I think, relevant to metabolism in this audience is building muscle. And so what we started making was a molecule called follistatin, which is basically the biochemical pathway for skeletal muscle exercise. And we could inject these mice and get like really profound results. So in this case, they're 6 months old, so they're basically just adult mice. We injected them once, and then we measured their grip strength and weight for a year, and then did a bunch of analysis on their muscles. But the general goal here is that if you think a person by the time they're 80 loses about 50% of their muscle mass, and physical strength becomes everything when you age, and it has a profound influence on your metabolism. And if we could make old people strong, we think we could improve quality of life and health span quite a bit, in addition to cleaning up a lot of these metabolic parameters. But our kind of first intended application is a prophylaxis for hip transplant. So before you got your transplant, you'd be treated with this. So you would be building muscle basically before you went into it, and then when you were immobilized, you would still be gaining muscle because your body thinks it's working out, but it's not. And the results in the mice were quite impressive. Single dose of this made them 50% larger and twice as strong in a year. And so I think that we could deploy this broadly. Like it's a simple infusion of these PLVs. It actually targets the liver, and the liver cranks this thing out around the clock. And so I think that this could have a massive impact on aging, longevity, and metabolic health. The other one that's kind of right down the center of the fairway for this is killing adipocytes. And we basically took that same suicide gene I showed you earlier that we're using to get rid of damaged cells and cancerous cells, and we turned it against adipocytes, fat. And we're looking at this in a couple of different lenses. Initially, starting in kind of rare lipidemas, abnormal like fat deposition diseases, and then moving into broader metabolic health. And the goal for this longer term is to be able to selectively target visceral fat. So you would not harm your subcutaneous adipocytes at all, but the kind of more metabolically chatty and pernicious kind of fat would be selectively ablated. And the first study we did in this is actually human tissue explants. So people would get like a tummy tuck done, and they take the excess skin and take little cookie cutters to it and send it to us, and we could inject the treatment into full thickness human skin, and then look at it for any signs of damage or efficacy. And in this early iteration of it, one injection killed about 20% of the adipocytes, and it didn't cause any harm to the surrounding tissue. And so this kind of encouraged us and led us to do actually a study that I'll show you right now. This is an in progress study, incidentally, so it's just something we're working on now, but we're taking obese mice and injecting them now locally with this, and just measuring the skin fold thickness. And so when this study is complete, we'll have MRI data and some other more detailed stuff. But we're basically able to reduce the skin fold thickness with this in a living animal as well. And so our plan is to take this forward, settle on an optimal formulation, and then take it through to regulatory. Just a couple of things to close, to inspire on the collaboration side. We can basically make any protein that you might want to make from pretty much any tissue in the body. And so if you guys have things that might be interesting to deliver, we're always interested in that. And then another fun project that's a little off topic but fun is we started making CAR T cells inside of patients. So instead of having to take your cells out to turn them into these cancer fighting cells, we could hit them in place and cause them to go after tumors. So I will stop there. I'm close to my 15 minutes and leave a little time for questions, but thank you very much.
Thank you, Matthew. Appreciate it. Remind us, what is the closest candidate for anything clinical?
Well, the COVID vaccine, although it's not Ocean's, is in clinic today. we've gone through the initial regulatory conversations with the cancer applications, but I think that it will likely be either a senolytic for kidney disease or the fat killing one. And the fat killing one is an earlier project, but its ability to go local administration only in a rare disease, I think, could give us a speed boost to clinic on it. So it's easier to pull off than the systemic ones. So that's I don't have a definitive answer, but we're having these conversations early next year. So.
Naturally, of course. And in terms of fundraising, where are you and where do you want to be in, say, 6 months to a year?
So we're going to do Series A at the beginning of the year to raise $30 million and going to push us ideally take the first thing through a phase 1/2 clinical trial and advance the other projects to kind of late stage preclinical where we have good packages. And so far, between Ossian and Senescent, like the sister companies, have raised I think about $15 million. And so we've been at it for a bit, and a lot has changed in the last year with Entos getting this big deal because they got a lot of money from it. It served as a lot of validation. I think, when we had started this, it was, I think viewed as a little bit crazy. There's a new kind of technology, and now it's been in people and has big pharma validation. So I would, in a perfect world, I suppose, raise $100 million and take them all into clinic at once. But I guess we're subject to the whims of the market.
Yeah, and that isn't quite the ride you expect it could be. Sometimes it's better to go in series than in parallel. But exciting technology, Matthew. We're delighted to have met it, and we'll keep an eye out for your progress.
Thank you.